It is my pleasure to introduce you to David. David Vetter was born in 1971 at the Texas Children's Hospital in Houston. After 20 seconds of exposure to the world, he was placed inside this plastic incubator bubble designed by NASA, shown on the right. David was born with a disease called severe combined immunodeficiency, or SCID for short. David was not the first person to be born with this disease, but he captured the world's attention and shed enormous light on this disease. David was placed inside of this plastic incubator to protect him from the outside world because he lacked very important blood cells that defend him. And because he lacked this cellular defense, he was placed inside these physical barriers as a spacesuit and the incubator to replace that cellular defense. Severe combined immunodeficiency, or SCID for short, is a rare genetic disorder that causes life-threatening problems with your immune system. If we look at blood draw samples from healthy patients, and we look inside their blood, we can see there are many different types of immune cells that help support and defend us. However, if we look inside the blood of patients who have SCID or look in blood draw samples, we can no longer detect these very important blood immune cells. In part because of this disease, every baby born in a US hospital has their blood tested. The only cure for this disease is to undergo a bone marrow transplantation to replace all of the blood cells in your body. One of these very important defenders is called T cells. We rely on an army of T cells to defend us from the outside world. And so T cells are a specialized type of white blood cell in our body that help attack foreign invaders, such as viruses, to keep us healthy. To fully understand the important role that T cells play in our lives, let's walk through the case of someone being infected with the flu. I'm sure everyone in this room is very familiar with the symptoms of the flu. Flu is a virus that infects your lungs, and so some T cells can recognize when a lung cell is infected, shown here by the cell nuclei in red. And so once these T cells recognize that there is an infected lung cell, these T cells can directly attach to the infected cell and kill that infected cell. And as a result, we are allowed to recover from the flu and no longer have infected cells. And so this is incredibly important, and I would also like to point out that through this interaction, this is a very directed and specific target, and that this T cell is attacking only the infected cell, but leaving the neighboring uninfected cells not harmed in this interaction. And so T cells are incredibly powerful, and we need them to recover from many infections. But what happens if a T cell can recognize and harm your healthy cells that are not infected? Now, that would be quite devastating, and as a result, we would develop autoimmunity. Autoimmunity is your own body's immune system attacking itself, and it can occur in many different ways. But I would like to focus on the autoimmune disease type 1 diabetes. So type 1 diabetes is an autoimmune disease where T cells recognize, attack, and kill a specific type of cell in our pancreas. And so shown here, we have our healthy pancreas cells, and then along comes a T cell that can recognize and attach directly to that cell and induce killing. And so this goes on to lead to repeated organ damage and becomes a lifelong disease. And so when our bodies are creating T cells, we have to think carefully about the fact that we need these cells because they're incredibly powerful, but they also need to be tightly regulated, and so they don't harm us. And so first, we have to have T cells, as they are that powerful source, without which we would have to live all in our own spacesuits. And as well as we need them to attack all our non-healthy tissue. And so with this criteria, how does our body create T cells? And so to understand this process, it is with my pleasure to introduce you to my favorite organ called the thymus. So the thymus is a small organ located above your heart that its sole purpose is to train this army of T cells to defend you. People born without a thymus do not develop T cells. And so the thymus plays a really critical role in our health by generating these T cells to defend us. And so stem cells from our bone marrow travel to the thymus where they become T cells. So all blood cells, including T cells, originate as originate in our bone marrow as stem cells. And once these stem cells arrive into the thymus, the stem cells are converted into T cells, and then after which point those T cells are trained. Once T cells are fully trained, they can enter our blood, and that's where they begin to defend us. And so the thymus uses these two steps that we discussed earlier to create T cells. It first ensures that these stem cells have in fact successfully converted into T cells, and second, that these T cells will not harm us, but will be useful to us. And so a helpful analogy that you can think of when thinking of the thymus, you can think of it as security at SeaTac Airport. So stem cells arrive from the bone marrow, stem cells arrive from the bone marrow to the thymus, just like people arrive from the airport. And you can think of these two steps by the thymus to develop T cells as the first two steps at security at the airport. So in step 1, in the ID check, it serves as a functional test for T cells to ensure that they have successfully converted from stem cells to T cells. And second, the screening of the luggage serves as the screening of T cells to ensure that they will not be harmful to us. And so in step 1, in going through security at the airport, you have to show some form of identification. But not all forms of identification are equal. I cannot walk up to a TSA agent and show them my UW student card and try and make my flight on time. So with that being said, you have to show a specific form of identification to the officer. And so this is similar in T cell development, and that a T cell must show some form of identification, and it must be the correct form of identification. So in this case, a T cell's ID is a protein, but there are many different proteins in the body, and so the specific type of identification that a T cell must show is called a T cell receptor. And that T cell receptor must be shown outward on the cell surface. Similar to the person here showing their ID card outward, it can't be within our pockets or within our bag. And so once a T cell has shown its ID or its T cell receptor on the cell surface, it has successfully passed step 1, and then that T cell can move deeper inside the thymus to undergo screening 2. And so step 2 of screening is focused on ensuring that no harmful objects make it into the airport. The security officer looks at their list of banned items and ensures that if they find any of the banned items in your luggage, that they will then remove them. All other items are allowed to pass through and enter the airport. And so this is similar to how step 2 works in the thymus, except cells don't have to take off their shoes or remove electronics. The harmful objects in this case are the T cells themselves, and actually identifying what these T cells see. And so a thymic officer cell is making sure that no T cells will recognize your own tissue and could lead to autoimmunity. So say if a T cell were to recognize that specific type of pancreatic cell, the beta cell, the thymic officer would remove it in the hopes of preventing you from developing type 1 diabetes. And so if a T cell doesn't recognize or harm your own tissue, then the thymic cell allows the T cell to enter our body and into our blood and begin to defend us. And so that T cell would successfully pass step 2. And so through T cell development, the thymus has created this army of T cells that not only work, but will now go on to defend us and not attack us. And so the thymus plays a really essential and powerful role in our overall health in generating this army of T cells. But despite this powerful role, the thymus is sensitive to many things, unfortunately. And this includes, but not limited to, medical treatments that we undergo, such as chemotherapy and radiation for cancer treatments, as well as stress, which I'm experiencing a little right now up here. And so this is how I chose this icon based on how I imagine myself at my desk sometimes, as well as poor diet is also sensitive to that. I'd like you to take a moment and think to yourself, how does an organ like the thymus show that it's stressed? So the thymus shrinks down in size. And so as of right now, my thymus is shrinking just a tad little bit. But don't worry, the thymus has an amazing ability to regenerate itself. And to understand just how powerful thymic regeneration is, let's walk through what happens when a 3-year-old patient with brain cancer undergoes chemotherapy. So one way to track thymic function in humans is to take a blood sample from a routine clinic visit and then analyze their blood for T cells. So shown here on the y-axis, we have our number of T cells that was within that blood sample, and then we can track their progression through treatment on the x-axis. And so we have our pre-treatment, so their T cell levels prior to undergoing chemotherapy. We have their cycle 1 and cycle 2 of chemotherapy, denoted here as C1 and C2. And then we have the months following chemotherapy post-treatment. And what we can see from this is that as the 3-year-old T cells are undergoing chemotherapy, we can see that they plummet, the T cells levels plummet during cycle 1 and cycle 2. But then over time, in the months following chemotherapy, we see T cells re-entering our blood and entering back in. And this is a result of the thymus is particularly sensitive to chemotherapy. And so in that time, we see during cycle 1 and cycle 2, the thymus shrinking down, and then it regenerating and being able to support T cell development, and we can see T cells re-entering their blood. And so this is a really amazing and powerful regenerative capacity. However, there's some things that the thymus can't bounce back from, which is unfortunately age. And so the thymus is at its max size when we are infants, and then it shrinks down over time. And so to understand this, let's compare the thymic regenerative capacity of that same 3-year-old patient with a 23-year-old patient with brain cancer undergoing chemotherapy. And so we have our same experimental setup where we are looking at blood draw samples from a routine clinic visit and then analyzing their blood for T cells throughout treatment. And so again, on the y-axis, we have our number of T cells. And then on our x-axis, we have our different stages of treatment. So first, we have our pre or baseline treatment prior to them undergoing chemotherapy. Then we have our cycle 1 and cycle 2 of chemotherapy. And then we have the months following chemotherapy. And so the 3-year-old T cell levels will be colored in blue, while the 23-year-old samples will be colored in red. And so what we can see from comparing the 23-year-old and the 3-year-old is that they start out at roughly the same baseline in the blood. Then they both plummet as the thymus is particularly sensitive to chemotherapy, and we see those T cells drop almost down to 0. And then we can see them begin to recover, and while the 3-year-old, after 10 months post-chemotherapy, will be roughly back to the baseline for pretreatment, that 23-year-old is unfortunately, after 15 months out of chemotherapy, is still working to recover back to that baseline of their T cell levels. And so this is significant because longer gaps without T cells means longer gaps without T cell help and T cell defense. And so going back to our airport analogy, this would translate into less security officers at the checkpoints, less screening, and potentially more harmful objects getting into the airport. And so the shrinking thymus as we age does correlate with increased risk of cancer, autoimmunity, infection, and decreased vaccine efficacy. But a smaller thymus can still be functional, though. An adult thymus, despite its smaller size, can still support T cell help, and that's very significant. And so a recent study where researchers and doctors at the Center of Regenerative Medicine in Massachusetts General Hospital followed over 1,000 patients who were recovering from heart surgery. Both groups shown here had heart surgery, but one cohort of patients, group 2, had their thymuses removed during the heart surgery, while the other group, group 1, retained at least a portion of their thymus. Heart surgeons routinely remove the thymus during heart surgery to gain better access to the thymus. Because as you can see in the picture shown here, the thymus rests directly on top of the heart, and so its very presence can get in the way in some very important heart surgeries, including and especially in infants. And so researchers and doctors followed the patients for up to 5 years after their initial surgery and compared many readouts between these two patients, such as their risk of death, risk of autoimmunity, and risk of cancer. What I find most interesting is that patients who had their thymuses removed, so the patients in group 2, were more likely to develop cancer and were more likely to develop many different types of cancer throughout this. And so all of this is to say that an adult thymus, despite its smaller size, still plays an important role in our overall health. So how do we better support the thymus as we age? And so currently there are no clinically approved medications to help the thymus with age or help us recover after cancer treatments. And so one of my research focuses is trying to develop treatments to help thymic function and to better support it with age. But before we can answer this question, we have to understand why the thymus shrinks and how it shrinks. And until we can understand the why and the how, we can't make meaningful change in designing treatments to help us. And so why is the thymus shrinking? And so we can return to our airport analogy and think of this as a huge labor problem. And so these thymic security officers, over time, some of them will just quit altogether, and that's how we can see that massive reduction in size overall in the organ, as well as some of them will decide that they're going to come to work, but they're not going to do any of the work to be there. And so we call these aged thymic cells, or you can also call them couch potato cells. It's my fun little name for them. And so they form pockets or scars in the tissue where no T cell development is occurring. And so to understand just how big of a problem this is, let's imagine that we have 10 security checkpoints. And half of them close due to those workers just outright decide that they're going to quit. And so we have about half, we have 5. That seems pretty good, right? But now 3 of them become aged thymic cells or couch potato cells. And so now we're only left with 2 security checkpoints. So that's 2 places where travelers can enter into the airport and only 2 places for T cell development out of our starting of 10. And so that's a pretty big problem. And in addition to them outright quitting or becoming couch potato cells, we are also now having some research to suggest that these young, hardworking thymic cells become jealous of their couch potato cells counterparts, and so they will all convert in neighboring areas to become couch potato cells, and we'll see these pockets that are devoid of T cell development. And so this is a really active area of research that right now I'm investigating in the lab and trying to figure out how we can better understand what's occurring here and influencing these healthy cells to become these aged cells. And so with our end goal of this is to design medical treatments that will act like a manager to whip the aged couch potato cells back into shape, and we'll keep them working to support T cell health over time. And so overall, to conclude, the thymus plays an important role in our health by generating these important T cell defenders. However, over time, the T cells shrink in size and form these pockets of aged thymic cells. And so we are actively trying to understand what's forming these pockets and how we can better design drug treatments to slow or stop the progression of these pockets from being formed, with our end goal is to support T cell development and function. And so with that, be on the lookout for more fun updates about thymic biology. And so this was going to be a fun 3D video that would rotate. Unfortunately, it's having some issues playing, but here's to the thymus, the most important and underappreciated organ in your body. Thank you so much for listening, and I'm excited for any questions at the break.